Okay, so I'm going to continue our discussion of extensions to Mendelian genetics. And I just want to review the idea of multiple alleles. And remember that this is where you have two or more alleles. So we have assumed thus far that there were only two possible alleles, and one was dominant and the other was recessive. We learned that sometimes those have incomplete dominance, but in this particular instance, this says that there could be more variation of a gene than simply two. So when we look at the example of the ABO blood type, when we look at the gene, it is gene I. And there are three different alleles of this gene. So there is a gene that is I with a big A. Let me erase that. I with a big A. You could have I with a big B, and then you could have the recessive. These two alleles are said to be co-dominant, which means that they are equally expressed. This allele, this little lowercase I, is recessive. And we would say that A and B are completely dominant over the recessive. So we don't know sometimes if we're carrying the little I. So we can talk about the genotypes and the phenotypes. So we could have this combination of alleles in our genotype. Notice that I still, even though there are three alleles, I still have only two alleles because we are diploid. So you could also have this combination, and both of these would yield type A blood. You could have this combination, and this would be type B. Notice that, oops, type B blood. Notice that the big B is completely dominant over the little I. So I actually do not know if I am type B blood, whether I have this genotype or whether I have this genotype. Then we could have AB. And this type actually refers to proteins that are found on the outside of the red blood cells. And the reason why blood typing is so important is that we need to be given blood that our bodies will not reject, so that the immune system will be able to accept it. So when we look at two little i's, this would be type O blood. Now this is what is referred to as the universal recipient. So it can receive any of the other types of blood, because what that means is that both the A and the B proteins or antigens are present on the red blood cells. So it's not going to detect it as foreign. And this is what is called the universal donor, because type O means that they have neither A or B. And so they would not be rejected if you have a type A person and you would transfuse them with a type O blood. Now this does not take into consideration positive or negative. And so when we talk about positive or negative, we can talk about what is called the Rh factor. And this is named after the rhesus monkey, wherein it was first discovered. So when we talk about the Rh factor, in this particular instance, we have positive, and that means that the Rh protein is present on the outside of the red blood cells. And this is coded for by Rh positive allele, and this is dominant. We have the negative, which means that there's no Rh protein on the outside of the red blood cells. And so this is Rh negative, and this is recessive. So for example, if I put these together and I talk about the inheritance of two genes, I could have this combination of alleles. So that is type O, And this would be positive because the Rh factor gene is completely dominant over the Rh negative. So this would be O positive. I could also have this combination. This is the genotype. This would be AB negative. Okay, so you should be able to figure out if I gave you this in the quiz or this in the quiz, you should be able to tell me what blood type they would be by first looking at the I gene and then looking at the Rh gene. Okay, so we are going to move on and we're going to talk about other exceptions to Mendel. And this is an example of where more than one gene determines the phenotype. So here we have gene interactions. So gene interaction. So let's go back to blood typing, and let's say that there is another gene that can influence whether or not the A and the Bs are put on the red blood cells. And so this is gene H. And this codes for an enzyme that attaches proteins to the outside of red blood cells. So big H is the normal enzyme, and little h is a defective enzyme. So this was first discovered because some people were having children that they should not be able to have in terms of blood typing. So let's use an example here of a cross. So let's say that we had a person that was this combination, and we crossed them with a person that was this combination. We could figure out what the possible genotype should be, and so we could put them into a Punnett Square where the gametes are on the sides. So this individual would have big A, and this one would have big A and big B. So I can put them together. So that would individual would have type A. This individual would have type A. This individual would have type AB. Oops, let's put the little b. And this one would have type AB. Right. So what would you say if these individuals had a child that was type O? So in the past, we might have used blood typing as a test for paternity, for fathering, but we now know this is not the case, because if a person has this combination, that's their genotype. It's the combination of these alleles that determine the phenotype. So the phenotype would be type O. The reason being that even though they have big A, that is not expressed because the enzyme that would attach those A's to the red blood cells is defective. And so we call this, or they have called this, it's kind of got a cool name, it's called the Bombay Phenotype. And that's when individuals are type O, even if they have a different combination of I alleles that should tell them that they should be otherwise. And so if you have a type O baby appearing in a Punnett Square or in a pedigree, and neither one, it shouldn't be possible, then this could be a possible reason. So in order for this to occur, the parents had to have been this combination. Because they're type A, they have to have one copy of the normal enzyme, and then they had to be carriers for that defective enzyme. Okay, they had to be, oops, that should be a B. So that's a B. So we can see here that the possible gametes for this individual would be this possibility. It could be this possibility. there's only 2 possibilities. For this one, there's 4 possibilities. So it could be IAH, big H, IA, little h, IB, big H, IB, little h, little h. So I could put these in a Punnett square. This should be H, not a B, so that should be an H. And so we could figure out then, for example, if I just look at this last one, you could have a possibility of 2 little h's coming together. So this would be, and I'm kind of messy here, but this is little h, that would be type O. This one would be oops. Oh, that's right. This one would be, and then little h, little h. Okay, so that would be type O too. There would be one more type O here, and it would be right here. So this would be big A, little h, little h. I think that's all the possibilities. There we go. Yep. Okay, so that's the Bombay phenotype. Now there is another really interesting example of gene interaction, or epistasis, as it's called. And this is eye color. So let me get to a new page here. And we are going to assume that when we're talking about eye color, that we're just going to assume that we just have brown and blue. So we have eye color. So we're going to kind of ignore green for the moment. So eye color is due to melanin, which is coded for by a gene, and this is a brown pigment. If there's lots of melanin, it will produce a brown-eyed person. If there's very little or no melanin in the iris of the eye, it will produce a blue-eyed person. So when we look at melanin, it is code for the eye is a gene called OCA2. So this produces melanin, and there's 2 alleles. So this is my gene. So I'm just going to use O. Okay, so this produces melanin. And little o, which I'm going to put a line over because it's lowercase, does not produce melanin. Okay. So we're just going to assume, for simplicity, that everybody can produce melanin. So we're going to assume that everybody is big O, and then therefore everybody that's big O should have brown eyes. So it's believed that humans up until 7,000 years ago were all brown-eyed individuals. And then we had a mutation that coded for a different gene that was a switch. And the gene that's a switch is called HERK. Let me make sure I get this right. So it's called HERK2. And this is the switch that turns the O gene on. So remember that we talked about lactase persistence and how there is a switch that can turn lactase on or turn it off. And in most people, it's turned off after weaning, but in some people, that gene is turned on through their entire lives. So they're lactase persistent. So if we go back and we look at this gene, and I know it's confusing, but I'm going to use H again because most people do. So this is a different H gene. So we have this H gene, and there was a mutation so that most people had the big H, and this is the normal switch, and the gene is turned on. And then we have a little h, which is the abnormal switch, and the gene is turned off. Okay, so there was a mutation that arose in the past where this switch got mutated, and it was a random, so mutations are random, and we're going to talk more about that when we get to molecular genetics. So that we have an individual that has this combination. Okay, so if this is this, then that switches off the genes, the O genes, and so this would be blue eyes. And so this explains the phenomena of people that have brown eyes and then have a blue-eyed baby. In the past, when we thought of eye color as being a simpler model, where you just had two alleles, one gene, that was not possible. But in this particular instance, it would be possible. So if we have a this combination, and let's say we have individuals that are of this genotype, okay, notice that they're both carrying that mutant allele, and then that mutant allele gets passed down, and so you can have an individual that is this. So this would be brown eyes, this would be brown eyes, and this would be blue eyes. So the parents had brown eyes, but they had a blue-eyed baby. So that is possible with this model of how eye color works. Now, the difference between blue and green has to do with the amount of melanin, but also has to do with its distribution in the iris. So the iris, I kind of think of it as like a mountain range, and the melanin can be distributed in different parts of the iris, and so it can give you different colors, ranging from blue to hazel to dark green. And there's probably another gene that controls the distribution of the melanin in the iris. And so it's more complicated. Eyes are more complicated than just a single gene. In this case, we're looking at epistasis. So this would be an example of epistasis because two genes are interacting to determine the phenotype. Now, there's been a lot of blue eyes in the media. And so if we look at some examples of this, let me get my Google up. Blue eyes, genetics. So this is an example of a website that says that blue-eyed humans have a single common ancestor. And so when they go back, they find that this mutation is thought to have arisen 6,000 to 10,000 years ago. And it is the cause of the eye color for all blue-eyed humans living on the planet today. And so you could ask, well, if I have to have two copies of this, and it only arose once, how could you get two copies coming together in an individual? And the answer is consanguinity. So in small populations, you might have distantly related, but related nonetheless, people who are marrying each other and having children. So it could be first cousins, it could be second cousins, it could be third cousins, right? We're kind of all related because we started out as a small population. So if you trace your lineage back far enough, you have some relatedness with the people around you. So consanguinity means the coming together of bloods. And in this case, it gave rise to blue eyes, which is not necessarily a bad trait to have. And if we look at the first ancestor that we think we have found that had blue eyes, this is a person that was living in Europe. They found his teeth, And they took the DNA from the teeth, and they found that his DNA coded for dark skin and dark hair, but his DNA also coded for blue eyes. And so you can look at this person and you're like, I think he was about 7,000 years old. And you could say, well, what would be the advantage of having blue eyes? Why did blue eyes become so common in the population? And they theorize that blue eyes were attractive. So they're kind of startling in a dark skinned person. So we can imagine that if you had a person that was blue eyed, you might be attracted to them. And so this is actually an example of what biologists call sexual selection. Okay, so sexual selection is that certain individuals with particular characteristics, oops, not selections, sexual selection. So certain individuals, like blue-eyed individuals, were more likely to reproduce and pass their genes on to the next generation because they were attractive to the opposite sex. So that's the news about blue-eyed people that has recently been discovered. So I have to go back to, okay. So continuing on, another exception to Mendel is what we call pleiotropy. So pleiotropy is pretty much the exact opposite of epistasis. And so here we have one gene causing many different changes in the phenotype. So genes can code for really important proteins, and if the protein is defective and it is important in a lot of different tissues, then it could have multiple effects. So an example of this is what we call Marfan Syndrome. And this is caused by a gene that codes for a connective tissue protein called fibrillin. And this is an important protein. And remember that connective tissue includes bone, but it also includes cartilage. And it's also found in the eye, and it is also found in your blood vessels. So connective tissue is found everywhere, actually. So when we look at the people that have Marfan syndrome, they have one defective gene, but they have many changes. So they have eye problems. They have an abnormal chest. So that would be like their sternum and their rib cage, and they have lung problems, and they have heart problems. So particularly the aorta might be problematic. They also have extra long arms and legs and fingers. And so you can see that there are many different changes in the phenotype, all based upon a single gene. So that is what we call pleiotropy. There is a famous person that is thought to have had Marfan syndrome, and that is President Lincoln. And so they predict that maybe he might have died of a heart attack. So he probably wouldn't have maybe lived to be an old age, even if he had not been shot and killed. This is another interesting example. This is a disease that is caused by a missing enzyme, and porphyrin will start to accumulate. In just a second, let me. Porphyrin, I'm sure that's right. Okay, so when we look at this is another example, which is called Porphyria. And so the porphyrins start to build up, and they specifically cause a problem in the nervous system. So they can cause dementia, So they can kind of cause, you know, crazy behavior, convulsions, being delirious, and then they can cause stupor where a person is not responding, right? It can also affect their muscles. They have weak limbs, and this affects their digestive system, and they can turn their urine a dark red. And so there was a famous person called King George, and there was a movie called The Madness of King George. And he was kind of a famous person in history because he would get really crazy and then he would start to act normal again, and then he would like have big problems with his digestive system. And so he kind of went from being crazy to normal to crazy, and they couldn't figure out what was wrong with him. And so then they discovered that after his death, much longer after his death, they don't discover it. They hypothesized that he had this particular disease called Porphyria. Okay. So the last thing I want to talk about in this video is what is referred to as polygenic inheritance. So this is a little like epistasis, but rather than just 2 genes, which we looked at, we can say that many genes determine the phenotype. Okay, so a good example of this is skin color in humans. Skin color can also be influenced by the environment because when we're exposed to sun, the skin will tend to produce more pigment, more melanin, in order to protect us from UV radiation, from sunlight, which causes genetic damage. So we're going to do, they think that there's maybe 6 or more genes that are involved in determining skin color, but we're going to do a 3-gene model. And I'm just going to give the letters to the gene as A, B, and C. So we have big A, big B, and big C, and these code for melanin. So they would all increase the darkness of the skin. And then we have little a, little b, and little c. So this is just a simplified model. And these would produce no melanin. Okay. So individuals could have this combination of alleles in their genotype. This is still diploid because I still have 2 copies of every allele. And then I can cross it with another individual that has this combination of alleles. So these individuals would be very medium skin toned, right? So they would just be kind of like middle of the road skin tone. And one of the really interesting things about this model is that if you look at the possibilities of the gametes, and I'm not going to write each gamete out, but I just want to point out that there can be an A and a B and a C, all those little alleles coming together into a gamete, and this one, and all those little alleles. So one possible outcome to this could be a person that has this combination of alleles. And so this is light skin. Okay, and the opposite is true. You could have a big A, a big B, and a big C coming together, so that you could have this combination. And so this would be dark skin. So 2 people that are of medium skin tone could produce offspring that are either dark skin or light skin, and that does not follow with the blending theory at all, right? So this is really important in terms of race because we are confused about race. And we don't realize that skin color is not a good indicator really of race. But sometimes we think that people should not be of a particular race because of their skin color. So if we look at the distribution of the skin color, this is a diagram that just shows what I talked about here. We have 2 medium colored individuals and they produce children. And this is the possible range of skin color. So they can have very light and very dark. Notice that most of them are going to be medium skin tone, so it's rare to have a lighter skinned and a darker skinned offspring, but it is in the realm of possibility. Most of them would be of intermediate skin tone. Okay, so this is a really cool picture that shows the mom and the dad and the two twin sisters. So these would be dizygotic twins. They would be fraternal twins. So they would be like sisters to one another. And you notice that they have dramatically different skin tones compared to one another, yet they are siblings. And then when we look at this picture, this is actually the aunt and the uncle to this child. And this is the Romas of Italy, and they are sometimes referred to as the gypsies, but that tends to be a derogatory term. And so a few years back, there was some concern because within the Roma populations, there was these very light-skinned children, and they thought that they had stolen the children and that they weren't true Romas. And so they were going in and they were removing the light-skinned children and taking them out of the families. And so when this is one child that was removed, and when they did the genetic testing, that they found that this child is the niece of these two individuals. So skin color was not a good indicator of relationship in that particular instance. And then you can have the opposite happening, and this is actually taken from a still from a film called Skin, which used to be something that you could watch off Netflix. And this is in South Africa during the apartheid in the 1950s. And so white people sometimes had children that were of a darker skin, but they weren't allowed to put on their birth certificate that they were white. And that prevented the children from being able to go to particular schools. They had to be segregated with the black people, even though their parents were white. And so this was a true story about a girl and her parents who went to court to try to fight that. And so this is an example of that polygenic inheritance giving us variation that we would not normally expect. Okay, so I'm going to stop there for today, and that would conclude these extensions to Mendelian patterns. So we talked about incomplete dominance, we talked about multiple alleles, we talked about epistasis, and we talked about pleiotropy and polygenic inheritance.